Okay. Okay, we're going to go ahead and get started here. Hopefully everybody can hear me. And we are going to pick up on the genetic code we were talking about on Monday. Anybody have any questions on anything to this point? Okay. So let's go into this. Put this up on the screen. Okay. Oops. So the last thing we looked at was this chart that shows you where the combination of the bases will give you the code for the various amino acids. Remember, there's only 20 amino acids. You've got 64 possible combinations of the codes. 3 of the codes are what we call stop codons. So they don't code for anything. They tell the ribosome, they tell the production of a protein to stop. There is one start codon, and that's for methionine. Over here, it's that golden AUG. That set of bases is always going to be at the beginning of every protein to start it. And so we really have, there's really only 61 bases. We don't have to learn the bases. You know, we don't have to learn the 64 possible combinations. We don't have to remember which one codes for what. You know, there's always a chart to tell you this. However, what is important is that we know how to identify what amino acid comes from what combination of bases. And so the combination of bases, the triplet code of bases is always going to code for a unique amino acid. So for example, if we had ACG is that's a triplet code, a codon. If we go down here to this first letter, we have A. And so we're in this row, right, the A row. If we come down the C column, that gives us ACU, ACC, ACA, and ACG. If we follow the G over, we can see that all 4 of those code for the same amino acid. See, we make sure that we don't have any mistakes. So even if we do make a little mistake, even if we substitute the wrong base in order for, you know, we want to make threonine. Even if we, you know, we need threonine in our protein. We need the amino acid threonine. Even if we have the wrong base for our third base, it still works, because threonine is obviously so important to us that all 4 possible combinations for it work. Proline has 4, serine has 4, alanine has 4, valine has 4, leucine has 6, and so does arginine. They have 6. So some of these are pretty common amino acids that must show up a lot in our protein. So we make sure we don't have errors in copying that by giving every possible combination that we can think of within reason to fit for those amino acids. So now we know what each combination of 3 bases will code to. So the order of the amino acids is dictated by the order of the base. The order of the amino acids in a protein is dictated by the order of the bases. So when we're saying this is the genetic code, what we're saying is, here are the instructions to make a protein. We have to have this amino acid followed by this amino acid followed by this amino acid in that particular order. And so when I say that the DNA is a cookbook, it's a set of instructions telling us how to assemble proteins. And since we are all about proteins, That's what it does. The DNA holds the information for everything about us by controlling the order of the amino acids that we use to put the proteins together. And that's, it's almost simplistic in how it works. And every organism uses this same approach for their DNA. Every organism, whether it's a plant, whether it's an animal, a fish, a dog, a cat, a human, a horse, a tree, a bush, a leaf, they all use this same approach to making their proteins. They all use these 20 amino acids in the order specified by their DNA. Okay. So we've got messenger RNA, which contains the order of the bases, which the messenger RNA is the clusters of the codons, which has the order of the bases corresponding to the order of the amino acids. Now we have to have a way to get the amino acid to the ribosome. See, because remember, protein synthesis, the actual manufacture of the protein, takes place in the ribosome. The messenger RNA will go to the rough endoplasmic reticulum. It will leave the nucleus, will go to the rough ER. A ribosome will form around the messenger RNA. Now we have a site of production of the protein. The third RNA, the transfer RNA, is going to be carrying each transfer RNA that's made will be transporting a unique amino acid. And that amino acid is specified by the anticodon on. Now, the anticodon is an area at the bottom of the structure of the transfer RNA. Transfer RNA is made up of nothing but a whole chain of nucleotides, with a base and a base and a base and a base. It could be uracil, it could be adenine, could be adenine, could be guanine, could be cytosine, could be adenine again, could be uracil in this funny shaped structure. Earlier depictions of transfer RNA used to describe it like a fish hook. Now the current thinking is it's shaped more like this with indentations on it. It doesn't matter. There's only two important structures on transfer RNA, and that's what we call the anticodon, which is a triplet. It's a triplet code of 3 bases here, and the area where the amino acid attaches. Those are the only 2 important areas on here. Now the anticodon down here, the 3 bases together in the anticodon, is the complement to the codon in the messenger RNA. So if your messenger RNA has a codon on it, this is mRNA. Your messenger RNA has a codon that has C, cytosine, cytosine. What is the complement to cytosine? It's guanine. So your anticodon that would show up on your transfer RNA that would be the complement to this would have an anticodon of GGG. So let's erase this and move forward. I think it'll get clearer here. So, okay. Translation. This is translation is taking the instructions in the messenger RNA and turning it into a finished protein. It's like taking a cookbook and following the instructions on how to make biscuits, or make a cake, or anything else. The as I said, the DNA is like a cookbook. The DNA cookbook contains instructions for every cell. It contains instructions for liver cells and skin cells and muscle cells and bone cells and so on. Every cell the information in the nucleus of every cell has all the information that any other cell does, but parts of it are turned off and parts are turned on. You know, if you're a liver cell, the parts on how to be a liver cell are turned on. The rest of it, you don't need the instructions on how to be a bone cell, but they're still there in the DNA. A cookbook, if you look up the instructions on how to bake a cake, how to bake a pound cake, for example, you're going to have all other recipes there, but you're only using the instructions for a pound cake. You're not looking at the other instructions because you don't need those other instructions, but they're all in the cookbook. You got everything in a cookbook. You got desserts and you got entrees and you got fried foods and things like that. You're only looking at the section on pound cake. And so in our bodies, if you take that analogy to our bodies, DNA is the cookbook. But if you're a liver cell, we're only using the instructions for the liver cell. So anyway, that is translation. Translation is when we are making a specific protein for a specific structure in the body. Maybe we're making hemoglobin. Maybe we're making eye color, hair color, skin color. Maybe we're making an enzyme. Maybe we're making amylase for our mouth so we can digest starches. Maybe we're making a neurotransmitter. Maybe we're making epinephrine. It doesn't matter. The instructions are all going to be specific. So the anticodon on the transfer RNA will actually bind to the codon of the messenger RNA. So, but only if it's complementary. So here we have a messenger RNA. There's our messenger RNA. And right here we have a codon. And the codon, for example, let's say the codon on the messenger RNA is AUA. Okay. What is the complement to AUA? Well, adenine is the complement to uracil, or uracil is the complement to adenine. Uracil is the complement to adenine here, and adenine is the complement to uracil. So the complement, this is the complement to the codon. This is the codon. The complement to the codon is the complementary pair. Adenine pairs to uracil, uracil pairs to adenine, adenine pairs to uracil. So this is the complement. So the transfer RNA, the anticodon, is the complement to the codon. And the anticodon is at the bottom of the transfer RNA. Now they will land together, they will join together at the messenger RNA in the ribosome. The anticodon and the codon will come together. Now remember, the transfer RNA is busy carrying its amino acid up here. So it's going to land on the messenger RNA. It's going to bind with the complementary codon. So the anticodons will bind to its complement on the messenger RNA. Now in translation, we have 3 steps, just like we had in transcription. We have initiation, elongation, and termination. So the first thing that has to happen is initiation. We have to find the starting point. Now, the initiator at the beginning of every section, every messenger, every code, every section of messenger RNA is going to have an initiator, and the initiator is going to be the codon AUG. AUG. Oh, stop it. Let me go back. I got carried away here. Okay, so let's start this right here and let me get it to. Okay, so at the beginning of every strand of messenger RNA, So here is our messenger RNA right here, mRNA. The first codon you're going to encounter is going to be AUG. AUG is the first codon on every strand. On every messenger RNA, it's always going to be AUG. This is the initiator. This is the start codon. The start codon is always AUG. Don't know why, but that's what it is. It's always AUG. Now, what will land on AUG is a transfer RNA with the complement. In this case, the complement, the anticodon is going to be have U, A, C. If AUG is the start codon, there's AUG is the start codon. What's the complement to A? That would be U. What's the complement to U? That would be A. What's the complement to G? That would be C. So UAC is the anticodon. The transfer RNA comes down and binds to the codon. The anticodon here, found on the transfer RNA, that's the tRNA right here. The transfer RNA is going to bind to the codon of the messenger RNA. Now, it's going to bring this very first amino acid. The start codon always brings the amino acid methionine right up there, methionine. So what's happening here? Well, as soon as the messenger RNA moves out to this rough endoplasmic reticulum, the ribosome will form around it. This is the ribosome. This is the large subunit and the small subunit. This is the large and small. This is the ribosome made of the ribosomal RNA. This is the messenger RNA. The ribosome has formed around the messenger RNA. The transfer RNA will come in and land at the complementary spot at the start codon. And it will always bring methionine as the first amino acid in every protein. Every protein will have methionine as its start codon, as its first amino acid. Now, there are two slots in the large subunit. And the first slot contains a transfer RNA that lands in the messenger RNA. There's an empty slot next to it. This is for the second tRNA to come down to here. Clear this out before I get any more confused on it. Okay, so let me go back and start this again. So we have our messenger RNA with its codon. It's the start codon. The ribosome is going to form around the messenger RNA, and a transfer RNA with the anticodon, the complement to the start codon, will land there. So here we go. There's the transfer RNA, there's the large subunit. Now notice that here at the second space, at the space available, we've got a, this is the second codon in the mRNA. Now, the second codon is GUA. What would be the complement to GUA? Well, that's going to be CAU. Cytosine is the complement of guanine, adenine is the complement of uracil, and uracil is the complement of adenine. So the transfer RNA that comes in and lands here at the A spot is going to look something like this. It's going to have an amino acid attached to it, and its anticodon is going to be CAU. and it will come in and land here and bind to this. Okay, so here we can start out. Here's initiation. There's the start codon right there. Here comes the small subunit. Here comes the transfer RNA with the initiation complex has formed, dragging methionine with it. There's our large subunit. Here comes our second one, arginine in this case. The anticodon is GCG. The codon is CGC in this example. Now, those two amino acids join together with a peptide bond. So we have a peptide bond forming right here. There's a peptide bond. Now here comes the second transfer RNA. It's carrying, the first transfer RNA is always carrying methionine. This was the start codon. Start codon. We now have we have our anticodon landing on the start codon. The second one has come in now, and the anticodon was CCU. The codon was GGA. They're complementary. We have a peptide bond that forms between the first and second amino acids. Now we're going to get a peptide bond that forms here. We're going to kick out the transfer RNA to make room for another one. Let's go back and, okay, so there is our start codon here. Here comes the anticodon for the start codon. Here comes the second transfer RNA to land here. The two amino acids joined by a peptide bond. The first transfer RNA is kicked out. There it goes. The ribosome slides down across the messenger RNA. Here comes the next transfer RNA. We slide a little further. Here comes the one coming after that. Notice how we can only hold two transfer RNAs at a time. We begin at the start codon. We land on, with the anticodon of number 1, landing on AUG, which the anticodon is UAC. The second transfer RNA comes in. It binds to the second codon. The two amino acids join together. We kick out the first transfer RNA. There it goes. The ribosome slides down across the messenger RNA. Here comes the third transfer RNA. And the first two and the third amino acid join with a peptide bond. The second transfer RNA goes out. When it leaves, it's going to go pick up another amino acid. And this process will continue. Whatever the codons are, the transfer RNA that has the correct anticodon will bring the appropriate amino acid. The transfer RNA will always bring the appropriate amino acid for the codon. The anticodon will tell you to, will be on, the anticodon on the transfer RNA will always bind with the correct codon specifying for the amino acid. And so we will build our chain, peptide bond by peptide bond with each amino acid until we hit a stop codon. Okay. Elongation. We will continue to build our chain. Every time we bring a transfer RNA in containing a amino acid, the anticodon of the transfer RNA will land on the complementary codon. The amino acids will bind together. The transfer RNA, the first transfer RNA that was in the first spot is kicked out and the next one comes in. And this will continue this elongation process will continue as long as we haven't hit the stop codon. We terminate when we hit the stop codon. There are only 3 stop codons: UGA, UAA, or UAG. The stop codon is on the messenger RNA. Just like the start codon was the beginning of the messenger RNA, the stop codon is the end. And so what will happen is, as we're chugging down the messenger RNA with the ribosome, going down the tracks of the messenger RNA, and transfer RNAs coming in and binding to their complement, the anticodon binding to the codon, once we get to the stop codon, everything stops. We see that here. So there's our stop codon, UGA. When we hit that one, that causes the ribosomes to separate, and it causes the messenger RNA to no longer accept any information, no longer accept any more transfer RNAs. UGA is probably the best known of the stop. There's only 3 stop codons. We've got UGA, UAA, and UAG. And down here we have UGA. That's the stop codon. As soon as we hit the stop codon, we stop making protein. The chain of amino acids linked together is released. Let me go back and show you that chain of. There's that chain right there. There's that chain right there that we've been building. Methionine and what's the next one? Arginine, and I think it's lysine's the next one coming up. There's our peptide bond. Here comes the next one. I think it's lysine, and they'll join with lysine. No, lysine, not lysine, lysine. And there'll be a peptide bond here. That one's gone, and this chain will continue until we hit the stop codon. And so there we're going along, and now we hit the stop codon, and oh, we didn't hit it here. We got to get one more. We hit the stop codon is UGA, and we stop. So however long the polypeptide chain of amino acids is, it gets released when we hit the stop codon. The messenger RNA will be, the nucleotides will be broken apart, ready to be reassembled. The transfer RNAs will go and pick up new amino acids just for its, specified by their anticodons. And now we have our protein. So we start within the ribosome. We start linking the amino acids together. We have the start codon for methionine telling us to start. And when we hit UGA, we stop. Now the protein then is captured in a transport vesicle and taken to the Golgi apparatus, where it is shaped and twisted and formed into its appropriate structure, and then gets released. Now, so from DNA to proteins, we use it depends on the complementary base pairing. The messenger RNA is a complementary copy of the DNA. The anticodon is a complementary copy of the messenger RNA. So we end up with the anticodon being the exact same sequence we had in the DNA. Consider this. There we go. Okay. Here's your DNA strand right here. This is DNA. This is the strand that's been opened up, and you're making a messenger RNA copy of it. Suppose your sequence here on the DNA is C, 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 G, G, C, T, T, C. There's your DNA sequence. Your messenger RNA copy, mRNA, your complement to that is going to be G, 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 C, C, G, A, G. Because remember, adenine goes to thymine and cytosine goes to guanine. So there's your messenger RNA copy of the DNA. The transfer RNA with the anticodons is going to be the complement of the messenger RNA. So now we're back to what's the complement to G? C, C, C. What's the complement to C? G, G, C. What's the complement to A? Well, in this case, it'll have to be uracil, which is equivalent to thymine, and C. Now, we are back now to the identical sequence. This sequence here is identical to the sequence in the DNA. So our transfer RNA is the perfect copy of the DNA. So we're still using the sequence, the exact sequence of the bases in the DNA. We've gone through, we made a complementary copy of the DNA, and then we made a complementary copy of the complement, if you will. The messenger RNA is a complementary copy of the DNA. The transfer RNA is a complementary copy of the messenger RNA, which means it is the same as the DNA. So the bottom line is we end up back where we started with the DNA. Now, problems with our DNA. We have mutations. Something happens with our DNA. One of those things we talked about that can damage the DNA, ultraviolet radiation or chemicals or pesticides or pollution or viruses or radiation, things like that. I think I said radiation twice. Okay, so there's our original structure in the gene, in the top one. We have, this is all DNA. And we have our original strand, the codon was CGA, ACC, CGA. But in the substitution mutation, which is a very common mistake, we put in, instead of having CGA, we have CCA. These three are these are the same, these are the same, these are the same. This is different. We substituted cytosine for guanine. Now, does that make a mistake? Does it make a difference? Maybe, maybe not. Maybe CCA codes for the same amino acid that CGA does, or maybe it doesn't. Maybe it codes for what we still call junk protein. What'll happen is that it won't code. If we have the wrong base sequence, it won't code. This is the kind of thing that our P53 gene is trying to fix for us by taking out the wrong base and putting the correct base in. And what we're seeing here is the sequence and is wrong. If our original sequence was CGA and now we get CCA, we've substituted a C in for the G. It may or may not be a problem. These things happen all the time. These substitutions, these are minor mutations that usually get fixed or the cell gets destroyed. How many places has it happened? We don't know. Do they all get fixed? I think so. Because remember, we have multiple locations for the gene. The gene isn't tied to one unique spot. There are multiple locations for it. Now, This is the substitution mutation that causes sickle cell disease. Look at this. Here we have normal sickle cell hemoglobin. This is the DNA strand here for normal hemoglobin up here. Here's the messenger RNA version of it. We have CAC, CTG, GAC, TGA, and so on. Until we get down here to CTC, glutamic acid. The messenger RNA complement of that is GAG. Makes perfect sense. C to G, T to A, C to G, glutamic acid. But here, we've substituted uracil in here. Actually, the mutation substitutes adenine for thymine. And of course, the complement is GUG. Now, what happens is, in sickle cell disease, the amino acid valine is put in at the number 6 spot. So we put it in at the number 6 spot, and now our hemoglobin, instead of having glutamic acid at the number 6 spot, we've got valine. And with valine, the hemoglobin is altered. The shape of the hemoglobin becomes brittle. It becomes twisted. It reduces the oxygen carrying capacity by about 80%. And this is a genetic disorder. You have to have sickle cell disease. You have to have both. They're caused by recessive traits. You have to have both recessive traits. But look at the similarity between glutamic acid, the sequence for glutamic acid, and the sequence for valine. Cytosine, thymine, cytosine, adenine, cytosine. The sequence is very, very similar. So not only is it one mistake at the number 6 spot, it's one base in the codon that causes this. So substitution mutations can be very critical. Sometimes they're not. Sometimes it doesn't matter. Sometimes we don't even notice it. Either we fix it or destroy it, or it actually codes for the same amino acid we wanted to begin with. But sometimes it's pretty significant. Sometimes we end up with something that damages our hemoglobin. And the reason that it's so significant is that if the hemoglobin loses its oxygen carrying capacity, particularly when we get stressed, then your patient's going to be short of oxygen. They're going to be unable to fight infections, and those brittle hemoglobin molecules are going to make the red cells brittle, and they're going to hurt going through blood vessels, because red cells are supposed to be squishy. Red cells are supposed to be squishy and squeeze through little tiny capillaries. They can't do that when they're brittle. They have a real problem. So one little mistake turns out to have some very significant consequences. Okay. Now, another type of mutation that's even worse than the substitution is what we call a frame shift mutation. This occurs when the DNA strands shift. They don't line up properly anymore. And we actually insert an extra nucleotide. So if you look at the top row, you can see the DNA strand broken out into its codons. The top row GCT, the other side of the DNA strand is CGA. Guanine and cytosine are complementary, thymine and adenine are complementary. You can see GCT and below it CGA. On the next row, next to it, you got AGT and its complement TCA. The third one, ATA and TAT. But suppose we insert an extra nucleotide on both strands of the DNA. The third codon is now altered. Instead of ATA, the codon on the top is GAT, and the codon on the bottom is CTA. that means everything slides down one nucleotide. So instead of the fourth codon being CGG, it's now ACG. And they're going to code for totally different bases, totally different amino acids. This destroys the gene. It isn't a little mistake like we have with hair color or eye color or skin color, where we're still going to produce the same protein, even though we got the wrong base in there. This is going to totally destroy the gene, and we're unable to make this protein anymore. And these frame shift mutations are very, very serious. Now, about our bodies, every cell, as I've been saying along, every cell has all of our DNA present. But all cells are going to be different. Cuboidal cells are different from columnar cells. Their location dictates what they're going to be doing. Some genes are turned on, some genes are turned off. All the information is there, but the cells are going to differentiate when we're growing in utero. They're going to grow into different things. They're not going to use the DNA that contains information on bone cells if they're a liver cell, or they're not going to contain the information on blood cells if they're a nerve cell. They're not going to use that. It's there, but they have differentiated into a different cell. They can be a blood cell, they can be a liver cell, they can be a skin cell. Whatever were predetermined to become is what they've become. They still have the information, but they're not going to use it. So now, problems with cells. Sometimes, we know that all cells except nerve cells and muscle cells are going to reproduce. Skin cells reproduce to replace damage. Scar tissue grows to replace damage in muscle cells, for example. We can have rapid cell growth called hyperplasia when we need it. And if we don't use certain cells, they can atrophy. Skeletal muscle cells, if they're not used with exercise, will shrink and get weaker. They will enlarge if there's lots of exercise, but they will shrink and get weaker. Cells also age. We don't know why. Cell lines, one cell divides, and that family line can divide 50 times, and usually after about the 50th division, it stops dividing. We think that cells age from wear and tear. They age from free radicals being developed. They age because our immune system gets weaker in those cells. But my personal favorite is the telomeres. That the telomeres at the ends of the chromosomes, the ends of the strands of the DNA, wear out. Every time we have a division, a little more telomere is lopped off. And once the telomeres are shortened sufficiently, the cell stops dividing. So now the telomeres don't grow. They only shrink it. Now in cancer cells, the telomeres will continue to grow. They will remain intact. They don't wear off, which is why cancer cells essentially are immortal. So my personal pet theory on this is the genetic theory, that it has to do with the telomeres being lopped off every time a cell goes through mitosis and cell division. And that is everything I want to talk about on the cell. So any questions on anything so far? Okay. Okay, we got a half an hour left, but I need to start setting up for my heart lab this afternoon for AP2. If you want to drop by and if you're on campus or nearby and you want to drop in and see some hearts being dissected and not have to wait till AP2 to look at it, you're welcome to come in. I do want to make an announcement that I will be offering AP2 as a hybrid class in May term. And so if you want to knock out AP2 before summer, before fall semester, you can sign up for that class. It is going to be a hybrid. So if you want to stay virtual on Zoom, you can do that. If you want to come in to the classroom, you're welcome to do that. You can go back and forth. We will be done with that course, but we will start on May the 10th, May the 9th, whatever the Monday is after graduation. I think it's May the 9th. We will be done by Memorial Day. We will go a chapter a day in AP2. Now, AP2 is much more interesting than AP1. And the amount of time spent in lecture is about the same as we would get with a night class. So you'll have 2 or 3 hours lecture followed by a lab. The labs in AP2 only take about an hour. They're much shorter than our labs, than the AP1 labs. So think about that. If you want to get rid of your AP2 over the summer, you can have it done by the end of May. And then if you want to take something else over the summer, like micro, for whatever reason, then there are some very good micro classes that you could take. And I think some of them are going to be are going to be hybrid. I don't know yet for a fact. But I know my class will be a hybrid class, meaning that if you like coming in virtually, like we're doing now, you can come in virtually. If you want to be on the ground, you can come in. But it's just an option for you to consider. So I'm not trolling for students. I'm just throwing this out there for you. So anyway, I'm going to end the class today. If you have any questions on anything, the summer class, exams, things like that, be sure to ask me. And I will, I guess today's Wednesday. I'll see you all next week.